This animation will describe the structure and contraction of striated muscles. All insect muscles are striated. Unlike vertebrates, insects do not have smooth muscles. The grasshopper femoral jumping muscle will illustrate the general structure of striated muscles. Muscles are comprised of many elongate cells called fibers. Fibers show the striated appearance and are multinucleate, indicating that they are derived by the fusion of multiple precursor cells called myoblasts. Packed within the fiber's sarcoplasm are contractile myofibrils. Myofibrils show the striated pattern as repeating subunits called sarcomeres. Sarcomeres are bounded by Z bands and divided into I bands, A bands, and a central H zone. Let us review the sarcomere at a more detailed structural level. The repeated pattern of the sarcomere results from the arrangement within the myofibrils of two major proteins, actin and myosin. Actin is a fibrous protein attached to the Z bands that are protein plates at each end of the sarcomere. The I band is a region of the sarcomere that contains only filaments of actin. The A band results from the presence of overlapping filaments of actin and myosin. The H zone represents the presence of only myosin. Let us now examine how a sarcomere undergoes contraction. When a muscle contracts, the actin filaments within a sarcomere slide along the myosin filaments. This has the effect of pulling the Z bands closer together and shortening the sarcomere. This view presents a series of sarcomeres similar to the situation during contraction by a myofibril. Let us now view the molecular detail of the actin and myosin proteins to demonstrate the molecular events that occur during a sarcomere contraction. This scene shows the molecular relationships of actin and myosin relative to the banding pattern of the sarcomere and demonstrates how this relates to sarcomere contraction. Note the relationship of actin and myosin molecules relative to the banding pattern that we have discussed for the sarcomere. Myosin filaments consist of crosslinks that bind to specific receptor sites on the actin. The crosslinks pull the actin along the myosin. Note that the crosslinks attach and release independently. If the crosslinks acted in unison, they would all be released at once, and the myosin would be detached from the actin, allowing the sarcomere to go back to its relaxed, uncontracted state. Let us look more closely at how the crosslinks slide the actin along the myosin. Myosin crosslinking occurs by a myosin head comprised of multiple proteins capable of conformational and positional changes. The myosin heads bind actin binding sites and undergo a conformational rearrangement in their proteins. The conformational rearrangement rotates the position of the head and pulls the actin along the myosin. In the next scene, we will look at the biochemical events related to actin-myosin binding. This scene shows the events that activate muscle contraction and the biochemical details of cross-linking and sliding of the actin filaments by the myosin heads. The cycle starts when an electrical impulse from a motor neuron stimulates a muscle fiber. Invertebrate animals may have 1, 2, or 3 motor neurons that control the contraction of a single muscle fiber, and each motor neuron has multiple end plate junctions spread over the fiber surface. The nerve signal from a motor neuron does not generate an action potential in an invertebrate muscle fiber, but rather depolarizes the entire surface of the fiber to produce contraction. The electrical depolarization also spreads into tiny membranous invaginations of the fiber membrane called transverse tubules or T-tubules. The T-tubules extend to the myofibrils. The T-tubules align with the A-band in invertebrate muscles. The T-tubules essentially carry the electrical signal into the sarcoplasm and along the sarcoplasmic reticulum, a complex of membranous vesicles that serve to store calcium within the muscle. Depolarization of the T-tubule membrane activates calcium release channels of the sarcoplasmic reticulum to release stored calcium into the sarcoplasm. The released calcium then initiates the muscle contraction by binding to a protein called troponin. Calcium-activated troponin causes a conformational shift in the protein tropomyosin, which winds around the actin helix and blocks the myosin binding sites on the actin molecules. This frees the actin molecules to bind the myosin head units. The contraction cycle starts with the myosin head bound to adenosine triphosphate, ATP. The myosin head contains adenosine triphosphatase, an enzyme that splits the high-energy phosphate bond of ATP to produce adenosine diphosphate, ADP, and inorganic phosphate. The resulting ADP and inorganic phosphate remain associated with the myosin head, which is also charged with the energy released by the splitting of the high-energy phosphate from the ATP. To start the contraction cycle, the myosin head attaches loosely to actin, then releases the inorganic phosphate, which causes tight binding with the actin. After tight binding, proteins in the myosin head undergo a conformational change that results in repositioning the angle of attachment between the myosin head and the actin. The shift to the new conformation pulls the actin along the myosin. After pulling the actin, myosin exchanges ADP for a new molecule of ATP. This results in release of the myosin head from the actin. The myosin head resumes its relaxed conformation and position, and the cycle is ready to repeat. Let us watch the cycle repeat several times.